Okay, sorry about that little break. Okay, so C-reactive protein. Basically, if actually, let's just start over on this side. We're seeing a tumor um, that is growing, and what started it all, we can see up top here, we've got chronic inflammation, basically creating that great environment for the tumor to grow. And then a downstream effect from the great amount of inflammation is that there's a protein made by the liver called CRP, and it basically is released to create more inflammation. So it's just this continuous feedback into the tumor. In the right hand side, we've got it in red, C-reactive protein, and they use this as a marker for cancer. It's been shown that higher levels, inflammation causes higher levels of C-reactive protein, and high levels, increased levels of CRP is correlated with a higher incidence of cancer tumorigenesis. Okay, so in this pathway, what we're seeing is CRP, the red thing, binds into this receptor, and then look what we see downstream from that receptor. We've talked about AKT before, and we've talked about mTOR before. So you can see that it does have the ability to interact with proteins that we know help tumors survive. Okay, on to the next hallmark. So reprogramming energy metabolism. First, let's review a little bit about cellular respiration. On your left-hand side, this is just a normal aerobic environment. And what we would see would be, so I'm going to draw an outer membrane, so that's a cell. And then this would be right here is the mitochondria. Okay, so what I'm trying to say, in the aerobic environment, the one with oxygen, the cells will bring in glucose and they will utilize it and break it apart into pyruvate. The pyruvate then will go through one more change, turn to acetyl-CoA, and will then go through the mitochondria electron transport chain. So what will end up happening is you'll make somewhere around 32 to 36 ATP. And that's the normal scenario when glucose is available, readily available, and there's a lot of oxygen. On the right-hand side, we're looking at a hypoxic environment, so there is not oxygen readily available. So what the biggest change we're going to see is that we need oxygen for the electron transport chain for it to continue going. So in a situation when there is no oxygen, then the electron transport chain isn't used. So the basic way it's going to maintain any production of ATP is by going through just glycolysis, which is this top part, and not the electron transport chain. Okay, so in this situation, glycolysis only makes 2 ATP. So not very good at maintaining a high production of ATP. So what do we see when we look at a cancer cell? Again, if we first start on the left side for glucose, this is a normal cell in A. Again, cell membrane, and then mitochondria. So we've got some glucose, little red molecules, and in order to move them through, we have to have a glucose transporter, so it shuttles in glucose, and with oxygen readily available, it will then go into the mitochondria, producing a whole bunch of ATP. It does still go through glycolysis, always does, but we're only producing 2 ATP. Okay, so totally you're getting a lot more. Now on this side is the tumor, again, cell membrane of the tumor cell, and then the mitochondria. What happens with the tumor cell is it changes how it utilizes an energy molecule. Specifically, in this case, we're talking about how it utilizes glucose. in the environment, remember those center cells of the tumor, that's not high in oxygen. So it has to reprogram how it's functioning on using energy molecules. So in this case, without the ability to make that efficient electron transport chain running, because there's no oxygen, you're not creating that high level 32 ATP molecules. All it can do is go through glycolysis. So it's only going through this stage, but it does increase the ability of how much glucose it can bring through. So it's got a much stronger glucose transporter system. So here over on the left side, we're seeing this one glucose molecule come in compared to how the cancer cell has been able to change the glucose transporter to bring in a lot more. So while it's still not going to make a ton of ATP, it at least is doing it at a faster clip of creating that 2 ATP. So that's one thing you're going to see. Another thing you're going to see is that it's going to release a lot of lactate, which actually plays into the other environments for tumor cells. Okay, so the Warburg effect is way back in the day. Otto Warburg came up with this idea that tumor cells are surviving using different metabolic programs. And as this material as research has continued to gain more evidence and material, we've been able to see this Warburg effect work hand in hand with other areas within the tumor. So on the bottom, we are seeing a cell that is only going through glycolysis. So we would say this is maybe more of those inner cells. It's bringing in a lot more glucose, in comes the glucose, and then we're creating a lot of lactate. Now, another set of cells within the tumor might be optimal for using that lactate. So it brings in that lactate using some enzymes, it turns it into pyruvate, and now you make more ATP. So it's just that this cancer cell can help that cancer cell. So it's very interesting to see how the environments will reprogram how the cell utilizes energy. Okay, and then our last hallmark, evading immune destruction. In this figure, we've got one type of tumor that has not been able to hide from our body's immune system. So we're seeing some apoptotic action going on. We're seeing some lysis going on. We've got other white blood cells that are actually binding to the tumor and recognize it, and that'll just keep pulling in more white blood cells. Now, if a cancer can figure out how to hide from the tumor or from the white blood cells, then it's going to survive longer. So in this figure, we've got a tumor that has figured out how to escape it. And then what's happening is it's inhibiting the other white blood cells that would normally come in, but it's able to evade being detected by those white blood cells. All right, so what's the proof that we have for this? On this page, you see a bunch of mice. These are mice that are immunosuppressed, so no or little immune system. Okay, so why is this important, and how does this show that if a cancer can evade an immune system, then we know it has a better possibility of become a larger cancer or a larger tumor? In these animals, they don't have an immune system. If you took a mouse that had an immune system and put a tumor cells into it versus a mouse that didn't have an immune system, put some tumor cells into it, if the immune system played a role at all, then the mouse without the immune system would obviously be affected. And that's what we see. We see that these mice that were immunosuppressed have a higher incident of cancer, almost like 100% they'll at points grow the tumor, versus the mice that have the normal immune system. Their immune system killed off the tiny cancer cells that were injected into it. So these are just mice that are bred specifically to test different cancer drugs, things like that, because you can't test cancer drugs first on humans. So first we do it in the cell culture, then we move up to the mice, and then we move into larger mammals like rabbits and things, and then we just keep going up. And they also so this is an animal model, right? And you can't readily immunosuppress humans, but we do know of diseases like AIDS that is immune suppressive, and those have a higher level of certain types of cancer. Same thing was seen, but not actually at a much increased rate, is people that have received a donated organ. And when you receive an organ, you have to be immunosuppressed so you don't reject that organ. But if that organ also had cancer cells on it and you're immunosuppressed, you could possibly gain the cancer from your donor. So that's been known to happen, but it doesn't always happen. So it's not like a for sure thing. Okay, and in this figure, oops, let's go back. This is the tumor cell right here, and it's just sending out a bunch of signals that cause an immunosuppression. So we're not going to bring in signals that would bring in white blood cells. We've got certain things. We've got Treg. So Treg, remember, that is a lymphocyte that regulates the T proteins, T helper, T cytokine, things like that. And so we're trying to bring down those white blood cells. Here, the tumor has killed a dendritic cell, so it doesn't activate the natural killer cell. The whole purpose of all these signals is to basically try to suppress natural killer cells, because we know those are the ones that find the tumor cells. So the tumor cells try to create an environment that is not hospitable and for the natural killer cells, or won't promote more natural killer cells or white blood cells to the area. Hold on. You're so close to being done, and then my presentation goes this way. Slideshow from current. Okay. Here, just another situation where you have a tumor and its environment, and what this tumor environment has to hide itself. One, it's going to release those immunosuppressive factors so that there are no white blood cells being called in. Here, we're going to try to stop T cells from creating more of the clones. Here we have not allowing active antigen presentations so that you're not calling in more of these white blood cells, and then again, suppressing more white blood cells. All right, and that actually finishes it up, since we went over the microtumor environment in the first part of this lecture. Make sure you're starting on your PowerPoint presentation. Remember that intro slide should be very basic, so somebody that's just the beginning bio major would understand, and then as you progress through it, you can get more technical. So first slide would be introductory, and then you're hitting a pathway, explaining that pathway, how it's related to that hallmark, and then a drug and how that drug actually works on the cancer cell. I have seen a couple of drafts of different presentations, so send me your draft if you want me to check it over. All right. Thank you so much for a great semester.